To this Fujifilm Cellular Dynamics and Nanion sponsored presentation. My name is TK Feaster. I'm the product manager for the cardio portfolio at FCDI. We're going to have two parts in today's session. In the first part, I'll talk about how groups are using iPS-derived cells in safety and disease modeling. And in the second part, Sonja will talk about the effects of CiPA drugs on excitation-contraction coupling and iCell cardiomyocyte squared. And this work will really be highlighting the CardioExcyte 96. And my part will really be just a primer, a quick introduction at a high level to get into Sonja's talk. So first I'll give you an FCDI overview, and then I will talk about a case study of drug-drug interaction with amiodarone and sofosbuvir. Then I will talk about a patient-derived model of hypertrophic cardiomyopathy, and both these studies are going to be using the CardioExcyte 96. And then I'll talk about some characterization of the sodium-calcium exchanger that we've been doing with the SURFR. And then I will introduce the iCell cardiomyocytes genetic diversity panel. So Fujifilm Cellular Dynamics is the global leader in the production of human iPS cells and iPS cell-derived types. We have two different divisions, a life science research division, which is really our tools business, and we also have a cell therapy research division. We have three main core competencies, which include iPS cell reprogramming, genome engineering of iPS cell lines, and then differentiating those lines into target cell types. We have over 900 patents, and we're based in Madison, Wisconsin, with two additional sites, one in California and one in the Netherlands. We have a significant global distribution network and a support network, and this support network really extends from technical support all the way to customer support. The FCDI life science portfolio has an array of different cell types. These include some neuronal types such as glutamatergic, GABAergic. We have dopaminergic neurons, and we also have motor neurons and astrocytes. We have a hepatic portfolio that includes hepatocytes as well as hepatoblasts, and then also our cardiac portfolio, which includes the cardiomyocytes and cardiac progenitor cells. And for this talk, we'll be focused mainly on the cardiomyocytes. So the iCell cardiomyocytes are really our flagship product. These cells have intact excitation-contraction coupling, and that ranges from electrical changes at the membrane being translated to intracellular calcium handling changes. And this includes calcium-induced calcium release. Then that calcium interacting with the myofilament, resulting in contraction, as displayed here as mechanical beating. These contractile properties can be picked up on this Nanion CardioExcyte 96 system. Here I'm showing just some baseline traces. One is for extracellular field potential on top, and the bottom is the impedance, which is really that contractile waveform on bottom. You can see both these have stable feeding, and there is a nice morphology for the traces. In panel B, we're just showing the effect of vehicle over time on cardiomyocyte function for beat rate, impedance amplitude, and impedance beat rate, and showing that there's no change with vehicle. So I just wanted to show a couple known pharmacological agents, showing Vandelanib, potassium channel blocker. That's a well-known HERG or potassium channel blocker, and you can see that control is in red, and Vandelanib at 5 nanomolar, 0.5 nanomolar, so 0.5 nanomolar prolongs the EFP, and that also prolongs an impedance signal. In the panel B, we're showing nifedipine, which is a calcium channel blocker. Nifedipine is going to shorten that extracellular field potential and reduce the impedance amplitude. And this is really just to show you at a high level how these cells respond appropriately to pharmacology. And I'll let Sonja go into more pharmacology in her talk and other known compounds. I also wanted to mention this amiodarone and sofosbuvir story. This work was published by Millard et al. In this, they're looking at sofosbuvir, which is a hepatitis C compound, and amiodarone, which is an antiarrhythmic, and they wanted to look at the effect of these compounds, their interaction on cardiomyocyte function, using the CardioExcyte 96. So here I'm showing baseline recording, just the stable contractile property of that waveform. And then when you add the compounds, both sofosbuvir and amiodarone, there's a reduction in the impedance amplitude, and there's also changes in the electrical activity, and that's quantified down here in panel B. Now we're going to transition a little bit and talk about a hypertrophic cardiomyopathy model. This is a patient-derived sample, so we took fibroblasts from a patient that had HCM. We made iPS cells, and we made those in the cardiomyocytes. This patient had a mutation in myosin heavy chain, R403Q. We compare this to a population control, so we're comparing the patient cardiomyocytes to iCell cardiomyocytes squared. Clinically, HCM can have preserved systolic function or enhanced systolic function, and it typically has diastolic dysfunction. So we looked at some of the parameters for amplitude of contraction, so this is really that force. And relative to this control, we showed that there's a reduction in that force. The beat rate is slowed. And if you look at the kinetics of contraction, so in that systolic interval, you can see the contractile properties are faster, and that there's an increase in the velocity. So there's enhanced systolic function, and the diastolic function is impaired. There's a reduction, and it's slower and prolonged, and that's consistent with the electrical activity. So we're able to pick up some of the in vivo phenotype of the enhanced systolic function and diastolic dysfunction in vitro using the iCell cardiomyocyte squared and the CardioExcyte 96. So now we're going to transition a little bit and talk about the SURFR. The SURFR is a transporter instrument used for transporter research. Here we're showing the N1 version. This has only one sensor. And then here's the 96 version. So this is more of a high throughput and this is more of a low throughput. Here's just an image of the sensor where you put your sample, so you can put your membrane or your cells here. And then there's an illustration down here showing the interaction between that sample and the gold surface. And the way this works is by applying different substrates, you can measure the current. And this is very good for picking up very low currents. So we wanted to take the SURFR and the iCell cardiomyocytes and see if we can investigate the sodium-calcium exchanger. This is not maybe one of the typical proteins you think about when you think about calcium handling, but it's very important. Here I'm showing a schematic of the intracellular space in pink, the sarcoplasmic reticulum, so that intracellular calcium is stored, is in white, and then the extracellular space and tubules are in white. When calcium comes out of the SR, it interacts with that myofilament and gives you a contraction. Then it's removed mainly back into back into the SR through SERCA. It's also removed to the extracellular space through the sodium-calcium exchanger. This exchanger lets in 3 sodium ions and 1 calcium out, and calcium can also be removed through this plasma membrane ATPase, but this one has much less of a contribution to calcium removal. Because the sodium gradient is so high, the sodium is rushing in, so this calcium is getting kicked out relatively quickly. So we wanted to evaluate that using the SURFR. And here I'm just showing some reason why one might be interested in the sodium-calcium exchanger. It has implications as proarrhythmia and also safety side effects. So we took the iCell cardiomyocyte squared in the SURFR and we compared and we did the experiments. As a control, we used HEX cells overexpressing the sodium-calcium exchanger. Here we're showing that current, so you can see both the HEX cells and the iCell cardiomyocyte squared have that robust current that can be detected with the SURFR. We increased the amount of concentration, showed a concentration-dependent increase in peak current. And when we calculated the Km, we showed that both have comparable Km, right around 100 micromolar, so the infinities were comparable. We then looked at some inhibitors, so here we're showing SEA-400, and as we increase the concentration of the inhibitor, there's a concentration-dependent reduction in the peak current, and that's also shown here in these representative traces. And then, surprisingly, we found that they have comparable IC50 right around 3 nanomolar. And then we looked at one more inhibitor, KBR7943, and as we increased the concentration of that inhibitor, there was also, again, a reduction in the peak current with comparable IC50 at around 5 micromolar. So this is just to show that we can use the iCell cardiomyocyte squared and the SURFR instrument and measure this current, modulate it with pharmacology, and do this in a high throughput manner. Now we want to transition and look at the effect of inhibiting the sodium-calcium exchanger on cardiomyocyte function. For these experiments, it's the iCell cardiomyocyte squared plated on the CardioExcyte 96 system. And we're using that SEA-400 compound at 2 different concentrations, 300 nanomolar and 1 micromolar. And in the representative trace, you can see that there's a shortening in this interval, so these cells are actually increasing their beat rate. That's shown graphically over here over time, so you can see there's a time and concentration-dependent increase. Baseline is in black, and then 300 nanomolar is in light blue, and 1 micromolar is in dark blue, so there's an increase in the beat rate. Now we're going to transition briefly. I'm just going to introduce our new iCell cardiomyocytes genetic diversity panel. This is something we recently released where we have iCell cardiomyocytes that are available from 30 healthy donors. So these are all cryopreserved, ready-to-use cells. The thought here is that one can take these cells and do functional or molecular assays, or one can select a population from this, and maybe they have a specific population they're interested in, and then do experiments and understand the effects of those compounds across different genetic backgrounds. A publication from Texas A&M was just recently published using this panel, so it's Griminal. You guys, you can check that out. I also wanted to highlight the demographics of the panel. Here we're showing again the males are in blue and females are in red. As you can see, the quantification over here for the panel demographics in the U.S. population. The panel has approximately 43% female and 57% male on the population. That's closer to 50%. The panel also has different ages, so it's kind of tough to see, but there's different age groups in case one was interested in different demographics or different ages. The panel has approximately 80% Caucasian, while in the U.S. approximately 72%. The panel has approximately 13% black, and the U.S. population is approximately 12%. And the Asian population of the panel is approximately 7%, while in the U.S. it's about 4.8%. So you can see there's nice agreement between the panel and the U.S. population. And even though not all groups are represented, we're still working on that. So that's my last slide. I showed you a little bit about the iCell cardiomyocytes and how you can use them to look at known pharmacology or unique drug-drug interactions. And I showed that with the amiodarone phosphorus story. I also showed you how you can use iPS-derived disease-specific models to look at in vivo phenotypes in vitro, and how that coupled with the Nanion system, you can measure that. And I also showed you how using the surfer, you can measure the sodium-calcium exchange in the iCell cardiomyocyte squared in a high-throughput fashion.
All right, so first of all, thank you, TK, for the invitation to make this presentation here. And I'm happy to show you guys today the CiPA validation study results on the iCell cardiomyocyte squared, which was performed with some non-core sites here. And just as a very brief and quick overview, yeah, I guess you've seen this a couple of times already. So the phase 1 pilot study was published like in April of this year. For the phase 2 data, we came up with the first non-core site publication in June. That's the box paper. And just right for the SPS meeting now, the Plinova paper came out for the core site study. And one thing to note here is that starting from the phase 2 study, like a combination of new technologies was being used amongst the different sites. And just for example, one of the systems was the CardioExcyte system, which is capable of recording field potentials and contractility via impedance. And the system is also allows to look at chronic and long-term recordings, which might be important for trafficking, for example. Also, people are looking at engineered heart tissues, or the cell optic device was being used, which is also in that paper here, and also some calcium imaging. So this was the setup for our study here. So we had 4 sites here, Roche, Novartis, and 2 of the Nanion sites. We all used a similar instrument, the CardioExcyte 96. We used the 4 iPS-CardioMyocyte cell lines, so iCell cardiomyocyte squared, core-for-you, pluricized, and cellatus cells. We did receive 23 blinded drugs from the CiPA consortium. We tested 4 concentrations according to the protocol with an N of 5 to 6 at each concentration. And again, also according to the protocol, here we looked at the acute effects, which is 30 minutes after drug exposure. This is what the CardioExcyte system looks like. So it's sitting outside of the incubator. It has this incubation lid here on top, so it's a controlled environment. We have 96 well plates, and basically the electrode design here allows to do combined recordings of both impedance and ESP. So you get some example signals here. So these are typical impedance signals, which are sort of a surrogate of the contractility of the cells, and the electrical properties are depicted as EFP signals. This is the set of compounds we received, and you see that different sites did receive a different combination of compounds, but for example, the high-risk compounds were tested at all 4 sites, and also according to the CiPA protocol, we were able to identify in the iCell Cardiomyocyte squared the different types of arrhythmia, so type A, B, C, D, and E, and we could capture these arrhythmia in EFP traces and also in impedance traces. And later on you will see these markers here, this one for type A, and this one here is pooled type A through E. You will see that in the table. Just one thing to show you here. So with the CardioExcyte, we do have an automated so-called secondary beat detection, so arrhythmia detection. So this is really automated. You see that in an online analysis. This looks like here, for example, with asfendisol, we get this kind of arrhythmia 11 or 17 minutes after drug exposure. And now for identifying which type of arrhythmia happened, we needed to manually go back to the traces and look and identify if it's type A, B, C, or E. Right, so these are the baseline values that we did get. So we had an average field potential duration of like 468 milliseconds, and the average beat rate, so it's all across all sites here, is around 34 beats per minute. And this graphic here basically shows the number of wells across all sites and experiments, and most of the cells here had around 0.5, 0.4, 0.5, 0.6 milliseconds as a FPD range. So these are the numbers here. So how do the cells behave in vehicle and positive control? So vehicle control looks very fine, so the cells did not do too much here. And for the fatty light, actually the different sites use different concentrations of the fatty light, but you do see a nice concentration response here anyways. So 0.5, 3, 5, and 10 nanomolar. And these markers here depict arrhythmia that was captured in the positive control recordings. This is one example of a low-risk compound. So this is tamoxifen. We did see a shortening of the field potential duration, a nice T wave. Impedance is not doing too much, so the contractility remains stable. The intermediate risk compound cisapride nicely was prolonging the field potential duration, and here a little bit of prolongation in the contractility at the APD value here. Then we have another example, high-risk compound disopyramid. So here we, for example, could see also a prolongation of the field potential duration, and clearly disopyramid is described as being a negative ionotropic compound, and we could capture this in the impedance data here. So you see a reduction of the amplitude in a concentration-dependent manner. This is the summary of all acute effects from all sites. So here are the high-risk compounds. intermediate and low-risk compounds. And for example, those trend lines here are basically a graphical depiction of the concentration response action. So if the line goes up, this means there's a field potential duration prolongation. And this was seen in all of the trials here. For the intermediate risk compounds, most of the FPDs were prolonged. And for the low risk compounds, it's the opposite. So you see the minimum is the blue dots here. This is the minimum point of the trend line. Also, the color coding here shows you in orange FPD prolongation of like more than 10% or more than 30%. So the high-risk compounds and the intermediate risk compounds mostly had the prolongation, and this would be minus 10 or minus 30% of FPD change. So the low-risk compounds were behaving accordingly. There were also the arrhythmia types identified here. So we have type A, or with this other marker here, type B through D. And so here you see the appearance of the arrhythmia at the very concentrations. So basically the minimum concentration which induced the arrhythmic events at the very experiment. So as a complete summary of that validation study for the CiPA, we found that none of the low TDP risk-induced drugs induced reverbalization, prolongation, or arrhythmic events in either cell line at around Cmax or up to 5 times Cmax. 6 out of 9 low-risk drugs did not induce reverbalization, prolongation, or arrhythmia-like events at any of the studied concentrations up to 20 or 240 times Cmax. And 3 drugs, like ranulazine, mexiletine, and metoprolol, induced arrhythmia-like events in higher doses in a portion of data sets. So in general, the low-risk drug effects in iPS cardiomyocytes were largely consistent with clinical safety records of these drugs. And actually these data are nicely matching also the CoreSight data that Xenia introduced already to you guys, probably. And the same is actually true for the summary of the high-risk compounds, so they are matching very good. So here the result is that all of the high-risk TDP risk drugs induced repolarization, prolongation, and arrhythmic events for us in 36 out of 36 data sets. And drug-induced arrhythmic events were observed at concentrations close to clinical Cmax for quinidine, Sotalol, and Vandelanib, and like 40 times Cmax for disopyramide. So again, the high-risk drug effects were largely consistent with the clinical safety records. And this is just a little bit of statistics here. So that's a cross-site correlation, field potential duration to beat rate. And for the iCell cardiomyocyte squared, we found a really good cross-site correlations amongst the 4 sites. And one thing to note here is that site 2, this is the purple one, used serum-free medium, and the others did use serum-containing medium. And so from this validation study, our suggestion for like a myocyte best practice would be, so if the cells show an expected and regular baseline based on FPD and beat rate, like this here, and if the positive control of the fatty light meets the required acceptance criteria, and also if the negative control values for every plate meets acceptance criteria, then the assay is predictive and reliable. So in total we found that the Myocyte CiPA assay on the iCell Cardiomyocyte squared is predictive and can be implemented in a drug development workstream. So what's next? We are currently drafting a Myocyte best practices manuscript. So together, so core-site and non-core-site people are working on that. We would love to submit this by end of the year. So this will be a nice guideline and hopefully bring more people to really use these cells for their assays, because it should be like a nice flow chart and like quick start guide for using the cells in the different systems. And also I really think that a deeper investigation of the already available, for example, contractility data from impedance, or like also comparison to other technologies like to 3D, like engineered heart tissues or the cell drum data. A comparison would be very informative and useful to really understand the pharmacology of the cells. So I'm just going to show you a couple of examples here. So this is the field potential traces in negative control and the impedance traces in negative control, and in the presence of 1 micromolar Vandelanib. Here you see arrhythmias in both the traces. And how does this now compare to the patch clamp data? So I really checked the HES ion channel data that we generated also in the other CiPA study, and tried to compare these results now. And looking at the Vandelanib data that were generated on a patch liner system, we found that for HERC we did get an IC50 of Vandelanib of around 0.23 micromolar. For sodium 1.5, the IC50 was around 9.9. And then we get the more IC50s for calcium, like 3.2, and Kv 4.3 for around 20 micromolar. And yeah, I think it's very interesting to see and understand that such a mixed ion channel block, having at a very low concentration here, lower than the IC50s here for some cases, is inducing these kind of arrhythmic events. Another example, this is Sotalol, so control, DFP and impedance traces. 10 micromole Sotalol is inducing these like rolling EIDs. And when we look at some synchrotron recordings here, we see that 10 micromolar is not inhibiting HERC too much, whereas, for example, 300 micromolar is doing a quite strong inhibition here. And the IC50 that was found was captured around 80.2 micromolar for HERC. And interestingly, so here 10 micromolar, which is much lower, is inducing these arrhythmia events without having blocked any of the other channels. So this is quite interesting to see. And I mean, the explanation that I have is that what you see here is sort of a very fast effect in a patch clamp experiment. And what you see here is a 30-minute incubation time. So I guess such a long incubation time with a synchronized beating cells is enough to like induce these arrhythmic events in a monolayer. So this experiment actually was done on a synchropatch system, which was equipped with temperature control. So the recordings were really done at 35 degrees. And just very briefly here, as an introduction of the system. So the synchropatch comes either with 1 or 2 modules, which are integrated in a Beckman robot. So you get like up to 768 cells recorded in parallel. You do get gigaseries, and most importantly, which is important for iPS cardiomyocytes, the system is equipped with a current amplifier and allows to control the temperature. And just one example here. So this is a plate where we had on the left side the iCell cardiomyocyte squared. In the middle we had HEC cells, and here it's shown NAF 1.7 cells. And for example, what you see is the stem cells are not capturing as good as the HEC and CHO cells, so the gray fields here mean that there is not a good cell captured on the whole, but still the success rate is quite nice and sufficient to do enough recordings. And these are some example traces here of the iCells. So we get a nice sodium current of a couple of nanoamperes in the different cells. Also calcium current is there. So this is like a double P protocol here. We activate the sodium current and try to have that inactive at this part to see the remaining calcium current. And this was all recorded in like a 2 millimolar external calcium solution. This is why it's so steep. These are some potassium currents with different kinetics here. This shape and the other shape, and mostly you have a mixture of both of these types here. And really, if I just take out one of these cells now and have a detailed look at the current the current density that the cells are showing. So here we have a nice sodium current, a calcium current here. And really, depending on the amount of calcium current you capture in that very cell, you get a more or less pronounced shoulder in the action potential when you switch to the current clamp mode. So mostly ranging like 200 to 300 milliseconds of an APD90 value. And this is how it looks like. So the other recordings were done on a patch liner. This is a single patch recording here, but they look equal. And you could tweak that a bit if you would use a dynamic clamp amplifier to introduce some more presence of IK1 current to bring the cells more negative and have the kinetics of IK1 present. But still, in that kind of mode, you can look at the action potentials that the cells just simply equip with the amount of channels that they have. So the summary is the iCell cardiomyocyte squared really performed great in the CiPA myocyte validation study. They are predictive. This is what we saw with all the CiPA compounds, looking at the repolarization data and the acute effects as suggested by the CiPA protocol. The CardioExcyte cell handling CiPA SOP is available. And also the iCell cardiomyocyte squared are suitable for automated patch clamp recordings in voltage clamp and current clamp. And also the cell harvesting protocol is available at FCDI or at Nanion. And like TK already showed, there's a functional evidence of the NCX transporters in the cells, and this is currently been investigated a bit more heavy at Nanion, because we think that this could be really a relevant target for safety pharmacology in the future. All right, thank you very much.